Ever wondered how proteins get their unique shape and function? This intriguing process is known as protein folding, a phenomenon that transforms a simple chain of amino acids into an intricate, three-dimensional structure capable of performing biological tasks. The journey of protein folding begins with the primary structure. This is simply the linear sequence of amino acids in the polypeptide chain dictated by the DNA's genetic code. It's like the blueprint of a building, outlining the raw materials to be used. Next comes the secondary structure. This refers to local folding patterns that emerge within the polypeptide chain. The most common patterns are the alpha helices, resembling tightly coiled springs, and beta sheets, which are akin to folded paper fans. These patterns are stabilized by hydrogen bonds that form between the amino acids. Then, we reach the tertiary structure. This is the overall three-dimensional folding of a single polypeptide chain. Picture a carefully folded origami structure, where every fold and crease has a purpose. The formation of this structure is influenced by various interactions, including hydrophobic interactions, hydrogen bonds, disulfide bridges, and van der Waals forces. Lastly, we have the quaternary structure. This level of structure is relevant for proteins that consist of more than one polypeptide chain. It describes the spatial arrangement of these chains and the interactions between them. These four stages of protein folding are driven by several forces. Hydrophobic interactions cause non-polar amino acids to cluster in the protein's core, away from water. Hydrogen bonds form between polar groups in the peptide backbone. Van der Waals forces are weak attractive forces between atoms in close proximity. Electrostatic interactions are ionic bonds between charged amino acids, and disulfide bridges are covalent bonds formed between sulfur-containing amino acids. During the folding process, proteins can follow different paths. In hierarchical folding, proteins progress through the primary, secondary, tertiary, and quaternary structures sequentially. The folding funnel model, on the other hand, describes how a protein explores various conformations before stabilizing in its native state. Sometimes proteins may require some assistance in their folding journey. That's where chaperone proteins come in. These helpers prevent misfolding or aid in refolding, facilitating the correct assembly of polypeptide chains. But what happens when proteins don't fold correctly? Misfolded proteins can lead to diseases like Alzheimer's and Parkinson's. Prions, which are misfolded proteins, can even induce normal proteins to misfold, propagating the problem. Scientists study protein structures and their folding processes using techniques like X-ray crystallography, NMR spectroscopy, and cryo-electron microscopy. Understanding protein folding is crucial in fields like biochemistry and medicine, as it influences the function and malfunction of proteins in living organisms. So, the next time you marvel at the complexity of life, remember that even the smallest proteins have an intricate story of folding and unfolding, a story that is still being written by scientists around the world.